Hereby I open this academic ceremony at Maastricht University. First of all, I welcome our candidate, Mr. Maurice Halder. He will defend in public his thesis entitled Dissecting Mechanisms of Heart Failure and in-stent restenosis using fate tracing and single-cell RNA sequencing. Welcome all members of the degree committee, and in particular the two supervisors sitting here opposite of me. Professor Leon Schurgers, he is Professor of Biochemistry of Vascular Calcification at Maastricht University, and Professor Rafael Kramann, he is Professor of Experimental Medicine and Systems Biology at the RWTH Aachen University in Germany. The 6 opponents I will introduce to you later during the ceremony. And I welcome you all here in the aula. Welcome to this ceremony, and I also welcome all the followers of the live stream. And I would like to invite Mr. Halder to present a summary of his thesis, and I wish you success in the coming hour. The floor is yours.
Thank you, Mr. Prorector, for the introduction. Dear members of the committee, dear family and friends, and dear colleagues also present, in the following presentation, I would like to give you an overview of the work presented in my thesis, entitled Dissecting Mechanisms of Heart Failure and in-stent restenosis using fate tracing and single-cell RNA sequencing. Nowadays, we are all unfortunately too familiar with cardiovascular diseases, which remains the primary cause of death, even unaffected by the recent pandemic. In my thesis, we focus specifically on heart failure, which also greatly affects our society and is one of the primary causes of hospitalizations. Further, its prevalence is expected to increase in the coming decades with our aging population. Now, heart failure can have many causes and forms, but it's universally marked by the characteristic that it can no longer pump enough blood around to meet the metabolic demands of the body. The heart, under continued pathologic stress, can remodel, which leads to scarring, or the fibrotic scarring of the heart, as you can see here. Now, this fibrosis scar itself is initially not a bad thing. It's kind of similar when you fall down and you cut your skin. However, the problem is that this scar does not fade away and is not replaced by functional heart tissue. And if the scarring of the heart becomes too severe, it will also limit the function of the heart, leading to heart failure. Now, if we talk about heart failure, we cannot exclude atherosclerosis, which is actually the leading killer of cardiovascular disease as a whole. Blocked arteries as a result of lipid buildup and inflammation in the blood vessel wall can also be a cause for heart failure. Now, to treat these clogged arteries, a procedure called stenting is performed. And it's also nicely illustrated here, as you can remember in the Ever Given incident in the Suez Channel 2 years ago, which is kind of like the coronary of all trade routes. Here, this blockage nearly caused a world economic failure. Now, to explain stenting a bit better, it's also depicted here in a more clinical setting. As you can see, the blockage is removed, and afterwards a stent, which is a small mesh wire tube, is inserted to keep the artery open. The main problem with current stents used in clinics is that about 10% of all these placed stents, in-stent restenosis occurs, which is the reblockage of the stented artery, needing for another stent surgery to reopen it, or in the worst case, this can result in severe complications for the patient. Now, the aim of this thesis was to focus specifically on the cells that drive these two conditions, meaning the cells that cause the fibrotic scarring of the heart and heart failure, and also the cells that migrate from the vessel wall and contribute to the blockage that we observe in in-stent restenosis. Because by gaining a better understanding of the involved cells and mechanisms, we can adjust our therapies to target specifically these cells that cause these conditions. Now, to explain a bit more what kind of cells we are investigating, I would like to explain to you what main cell types can actually be found in the arteries that transport the blood around all across your body, including to organs like the heart. So if we start with the inner layer of the blood vessel, we can find the endothelial cells, and these are in direct contact with the blood that rushes past with every heartbeat. Next to that, in the medial layer, we can find vascular submuscle cells, which are contractile cells which can contract or relax in order to regulate blood flow. Lastly, in the more perivascular, which means outer region, we can find a variety of fibroblasts there. And in chapter 2 of my thesis, we already described in our literature study that these cells can undergo great changes in disease. And however, still a lot is known about the exact contribution of each of these cell types. And since the perivasculature also harbors many cell types, we need specialized techniques to study them. In my thesis, we made use of two main techniques, which I would like to explain to you also a bit more detail. First of all, we use genetic phase tracing, where we can tag a specific cell of interest and follow them in disease. To do this, we can label any vascular cell or perivascular cell by injecting tamoxifen in these special mouse strains we have in our lab. This activates a red tag in these cells, and we can further enhance this red signal using microscopes that use lasers, and therefore a red signal is reflected. So we can kind of compare these cells to the reflector you can find on the back of your bike, and we can use them to follow them down the road of disease progression. Now we can make any cell type of the vasculature reflective, as I mentioned, right? Endothelial cells, VSMCs, and also these outer adventitial fibroblasts. And even when these cells would divide by division or become activated in disease, they would keep this tag. Also, if a cell would switch to a different cell type, it would also keep its tag, allowing us to really track these cells in disease and pinpoint their origin. Next, we also combined our phase tracing with gene analysis tools to see what kind of cellular mechanisms are really become activated in these conditions. Now, beforehand, we would isolate cells from our tissue and extract the gene expression data more into like a general pool, right, which would give us an average expression of the tissue, which we can compare to a smoothie, right? We know that there is fruit in there. However, afterwards, it's very difficult to say how much of each fruit type contributes to the drink as a whole. Nowadays, with the advent of new transcriptomic tools called single-cell RNA sequencing, we can actually keep our single-cell resolution and isolate the gene expression on a per-cell basis, which then resembles a bit more, I think, a fruit salad. We can see that there are many, we can see actually what's in there, how much of each fruit type is in there, and afterwards we can also pick each piece individually and study it in more detail. Now in chapter 3, we combined these two techniques in our mouse heart failure model. And to mimic heart failure in these mice, we inserted a constriction or narrowing of the aorta, as you can see in this picture, which induced a pressure overload of the heart. Now, as you can imagine, this constriction puts the heart under a lot of pressure, as now more contractile force is needed to pump enough blood around to meet the metabolic demands of the body. As a result, the heart remodels and also becomes scarred. So in these mice, we tagged our cells of interest and isolated them from disease and also from our control healthy hearts, for a combined total of 77,000 cells, which on average more than 2,000 genes associated to each cell. As you can imagine, this is a lot of data to process, as we're now dealing with very multidimensional data sets. And just to give an indication of how we analyze this, here's a visualization of all these cells, where every small dot, it's almost impossible to see, but every small dot is a single cell with more than 2,000 genes associated with them. Now, in our analysis, we initially focused a bit more on the fibrous population because they had the strongest gene expression signature associated with the fibrotic scarring of the heart. So from all these fibroblasts in our data set, we observed that there was a specific fibrous population which we termed ECM fibroblasts, which increased in numbers in our disease model. And by analyzing the gene expression data of this subpopulation, we found that they strongly upregulated a protein called THPS4. And since we also preserved the upper half of the heart for more histology analysis, we could also stain for this protein in our hearts, basically, that we had left. We found that this THPS4 in ECM fibroblasts was not expressed at all in healthy hearts, but in disease, strongly associated with these regions of injury where also the fibroblasts were located. We concluded then that this ECM fibrous population derived the fibrosis observed in our model, and also by diving a bit into the mechanisms of this, we found that there was a transcription factor called TET1, and in our validation studies in cell culture and also further stainings, we found that it was a regulator of fibroblast activation. Lastly, in this study, we also observed angiogenic changes in the angiogenic populations of our data set, meaning the cells that are involved in the formation of new blood vessels. As in our model, due to the increased stress, the hearts also became hypertrophic as a compensatory mechanism of the heart. Now, this increase in heart volume also increases the metabolic needs of the heart itself, which could be met by additional blood vessel formation. However, we did not observe this in our data set and in our validation studies, and rather found that there was only a more minor angiogenic response, which we deemed also to be insufficient and likely contributed to the scar formation observed in our model. Next, in chapter 4, we analyzed the process of in-stent restenosis. And this study basically had two arches. One where we performed the genetic fate tracing in mice with custom-designed stents that were small enough that we can plant them actually in these small mouse arteries. And then we could also perform the fate tracing, right? And secondly, in dyslipidemic rats, we placed clinical-grade stents, allowing us to collect enough tissue to analyze the mechanisms of in-stent restenosis using a single-cell RNA-seq technique. So from our fate tracing data, we observed that there was a strong migration of VSMCs from the middle layer through the stent struts, as you can see here. Now, this was already reported by other groups in the field. However, we could, other groups in the field, and they actually stated that this was one of the dominant cells that drive this process, actually. However, by using our fate tracing technique, we could demonstrate a clear contribution of GLI1 adventitial fibroblasts that migrate all the way from the outer perivascular region through the stent struts, also greatly contributed to the blockage observed in in-stent restenosis, thereby proving that this process is not dominated by VSMCs, but also strongly affected by adventitial fibroblasts. Of course, we were then very much interested into the mechanisms that could drive this condition, and unfortunately, we cannot perform this technique in the same mice that we use for fate tracing due to the low cell yield. However, by implanting them in our rat stent model, we also could mimic the situation in the patient a bit more. By analyzing our single-cell data using bioinformatics tools, we observed that there was a communication between the endothelial cells and the fibroblast in our restenosis samples. By validating some of these communications in our in vitro model using a human renal artery, we could validate that this afferent axis, which was reported also in fibrosis and inflammatory-related processes, that here on our experiments, it was an activator of fibroblasts, adventitial fibroblasts, and also caused their migration. And therefore, we think that this can be of great interest in future stent therapies that's focused specifically more on the cells that really drive this condition. Now, to conclude my presentation, I would like to summarize the key findings of my thesis. So in our heart failure studies, we observed there was a specific ECM fibroblast population that becomes activated in disease and starts upregulating also this THPS4 protein and drive the cardiac fibrosis in our heart model. Mechanistically, we found the transcription factor TET1 to be a regulator of fibroblast activation. And lastly, we only observed a minor angiogenic response, which we deemed to be insufficient to cope with the increased metabolic demands of the heart, and therefore contributed also to the fibrosis formation that we observed in our model. Lastly, in our in-stent restenosis studies, we found that by fate tracing also these perivascular fibroblasts, that they strongly contribute also to the restenosis observed in our model, and that this process is not dominated only by VSMCs, but actually more in tandem with these perivascular cells. Lastly, in our single-cell RNA-seq analysis, we observed that this EFMB2, EPHB4 signaling axis can be of great interest as it is an activator of perivascular fibroblasts and cause migration, and therefore should be studied in future research to develop more cell-specific therapies to combat the restenosis observed in patients. Now, with that, I would like to conclude my presentation, and thank you for your attention, and I give back the word to the pro-rector.
Thank you, Mr. Halder, for your clear presentation. The opposition will be opened by Professor Judith Sluimer. She is the chair of the assessment committee, and she is professor of cardiovascular pathophysiology at Maastricht University. Professor Sluimer.
Thank you, dear pro-rector. Dear candidate, it is really a pleasure to be here, also to read your thesis. We've had many interactions where you were mulling things between Aachen and Maastricht, and I thank you for that, but it was a bigger pleasure for me to read your thesis. And as you can understand, the topic is very close to my heart. I have to say congratulations to the team. Also, your publication list that well exceeds the content of this thesis is very impressive. So now let's see, after all the applause, talk a little bit more about the content. Well, candidate, I think you are aware that your promoter has received many applauses for all his omics and big maps, etc. But what I hear mostly from my colleagues is that they're a bit disappointed by what the single-cell sequencing actually brings. I think maybe possibly because people are not taking it yet as much to the next step. You try a little bit, but it needs a bit more functional causal studies following up and showing that all this description really means something. So I would like to discuss with you if you have any ideas to take your work further. Specifically, I like to talk about the stents. They're coated with certain drugs in clinic. So with the knowledge you now have, do you think these agents like sirolimus work for those new cell types involved, or would there be a better coating?
Highly esteemed opponent, thank you for your kind words. And for your question, of course, I agree. The field of single-cell RNA sequencing has been quite new. We were quite fortunate in our lab to be one of the first to use this technique. And this is a very evolving field, right? So new techniques are being developed. And in our studies, we also specifically focused on to really validate some of our findings, to also prove that what we see can actually be of help. Regarding your question about the stent coating, so indeed, current stents are coated with everolimus, which is more like this general antiproliferative agent, which blocks basically all cells, right? And we know also from clinical data that when the endothelial cells are blocked and stopped or hindered in their migration to cover these stent struts, this can also be of great damage to the patient, right, with late stent thrombosis, for instance. So as I mentioned in my discussion, I think the future really lies in more targeting both the VSMCs and these adventitial fibroblasts to really stop their migration, right? And we can kind of let these endothelial cells do their work and cover the stent struts. And if we target these more mural cells, I think this would be a lot better. And then we can hopefully reduce the restenosis rates observed in the patients, actually.
And do you have any molecules in mind that would have to address both the muscle cells and the fibroblasts?
For both, I think it's a little bit difficult. So, I mean, they are a little bit part of the same lineage, right? There are studies that I described this also in my discussion that really target VSMC specifically. In our lab, we have experience with GAN61, for instance. We also did some experiments with this. But I would focus morely on these perivascular fibroblasts because, and also in our data, we see that these are the first responders. We see an increase after 2 weeks, more that these Gli1 perivascular cells migrate firstly, and then it's more supported by VSMCs, right? So I think targeting the perivascular cells first can be of great importance. Yeah.
But how?
Yeah, so
I still have not heard a real molecule or a compound.
Yeah.
I want you to be more specific.
Yeah, so this was unfortunately a little bit beyond our scope. We are planning, of course, to also. So this work hasn't been published yet. We are trying to increase also more samples to really strengthen our single cell data. So I mean we looked into the into bioinformatics data, and we checked also these data sets, which have tier 1 druggable drugs, tier 2, tier 3. So there we should find probably one, and then we can potentially coat one of these stents in our own lab, and then see if this really inhibits the migration. Yeah.
And do you think that, if I'm not mistaken, TEAD1 was also the one in the in-stent restenosis, right?
This was from our heart study.
Sorry.
Yeah.
Okay. Then that goes to the next question. You already mentioned about the shared lineages, right? And I think it's quite complicated these days. If you look in the literature, nomenclature, whatever people call it, it's a fibroblast, fibroblast-like cell, pericyte. It's very difficult. And I was wondering whether or not you were aware of the work of the group of Mike Majeski and Mary Weiser-Evans. And if not, I can slightly...
It would be great if you could elaborate.
So they have studied cells that are SCA1 positive. With a mice in the 11 reporter, they show that they derive from smooth muscle cells, but they de-differentiate. So they're de-differentiated smooth muscle cells becoming SCA1 positive, Ly6CA positive. And recently, they actually found that these cells are actually expanding on injury. Mm-hmm. And even in some bioRxiv paper right now, they show they use Gly1 actually to mark those cells, because they say that that's the main, or that's at least increased expressed in those adventitial progenitors that arise from de-differentiated smooth muscle cells. So, I'm losing the plot here, and I was wondering whether you have any idea whether your data, how your data aligns with this work.
Thank you for this question. It's very interesting to see that you mentioned that they would de-differentiate back to this Gly1, which I think is very unlikely. I mean, from our phage tracing, really observed that they come from the outer layer and actually migrate in, right? And also looking at these images, I don't really see like a differentiation back, right? So I think this is a very interesting. I have to look at this paper, of course. I mean, the SCA1 indicates a more mesenchymal status of these cells, but...
No, I'm happy you agree, because I also don't really believe it. I mean, they de-differentiate, they go back again. Yeah. I mean, come on, I know they're very plastic, all these cells, but maybe we should write a review and then and set the nomenclature right, and you can guide us with the basics of your work. Yeah. I'm very happy with the discussion. I'd like to give the word back to the prorector.
Thank you, Professor Sluimer. The opposition will be continued by Professor Willy Janen-Deschent. He is Professor of Biointerface Science at the RWTH Aachen University.
Thank you, Prorector. Actually, I'd like to start off with a question that is maybe off the record. Candidate, can you go one slide back? I was wondering what the black piece on the left margin was all the time. Is that a fault in the projector, or is that the reverse of the right side of your artery?
Highly esteemed opponent, thank you for your comment on my presentation. If you have a look at the nice thesis cover I made here, you can see that this edge is this is the same edge used in the presentation. And as my opening slide starts with the cover, it actually shifts to the side, revealing this part. Okay. So I thought this was a nice inclusion of my
relieved to hear that the University Maastricht projector is okay.
There are some other issues, I think, with it, but no, thank you.
Candidatus, I would like to start off with figure 1 of your beautiful PhD work. I must say I congratulate all of your supervisors and also the family who've sent this very able candidate. And it's a pleasure to read through, and I could read through 10 times and still only get half of it, maybe. So let's start and then finish data-wise with figure 1. That is a picture of Gly1 positive cells on a bifurcation of a carotid artery. When you look, the cells are patchy. They are distributed very uneven. And if they are precursor cells or whatever they are, there must be a reason why they are arranged the way they are. Also, if you look closely at the bifurcation itself, there is no precursor cells. I wonder, and I mean that in the broadest sense, if you could speculate why these cells dwell where they dwell.
Yes, thank you, highly esteemed opponent. Regarding the patchiness, I also wonder if during the imaging process everything is captured as it is in vivo, right? So this was using confocal imaging, and I wonder if like all the cells actually are nicely visualized. If this is the case, as you suggest, I agree that during in the area of the bifurcation there might be less cells, right? And I think This can also be due to a different tissue structure observed in these areas, right? As you can imagine, straight after the heart, there's the aorta, and what we observe usually in vasculature is that there's certain areas of shear stress or increased shear stress, which also are usually these initiation spots for atherosclerosis or vascular calcification. So it could be that these parts are just on a functional basis have then maybe less Gli1 perivascular cells there located.
Mm-hmm. I agree. I mean, if they're precursor cells or even stem cells among them, they will have what it's called the niche.
Yeah.
And that must be constituted by something around them.
If I may add to this, the Gli1 adventitial fibroblasts are a subset of all perivascular cells, right? Mm-hmm. There are also other fibroblasts there which are not marked by Gli1, so it could also be that there are different subtypes there actually that are not visualized there.
Moving on to the kind of data analysis that you did, that is really, really great and full of data, and I'm sure you still have a lot to analyze in these data. You actually founded a company based on all the data that you have collected. So data collection seems not to be the problem anymore. It is data curation maybe, and then making sense of it all. With the advent of artificial intelligence, everybody hopes the more data we collect and the more intelligent we have machines to sift through the data, we come closer to a push-button solution that will make us sort of superfluous. But how far are we down that track? How far are we that we actually get away from the association, which we mainly study now? So this thing is blue, it must be sky, to causation.
Oh. Thank you for this very great question. I agree. So I think we're getting close to also like single-cell GTP, where we can use these techniques to annotate our subpopulations. However, in our work, and then of course my colleague Fabian, we dove into this data, and as I agree, you have more data than you can actually analyze, right? So we picked a few things, and also to test this a bit. But I think the bioinformatics tools still have like a long way to go, and really also translating it more to a functional meaning that we can actually test then. So I agree. I mean, and nowadays we of course as biologists we know a little bit. We know how the mechanisms work, and then we have to communicate this also to our collaborators, right? In our team, we also have excellent bioinformatics, but we need to bridge this link because they know how the methods work, but they kind of miss the more biological inference. So at the moment, we do this from both teams, and we work together on this. But in the end, I agree, it would be great if we have like a more AI-based approach, which would also mean that we are a little bit more unbiased, right? Because normally we find something in a data set which we are familiar with, and we focus on this. And if we actually start looking a bit more at what the AI might suggest, you can actually find something that's also unique and hasn't been discovered before.
Keeping in mind that the AI will necessarily be biased towards what is known before, because otherwise you can't train it.
True.
Which is another catch in the thing. But can you see from your data, for instance, when cells truly differentiate, like in embryo life, versus selecting cells, because they're the ones that fare better in the conditions? Can you see that in quasi-time?
So in our studies, we didn't focus on developmental biology. So I think it's, for me, a little bit difficult to comment on this. However, what we could analyze was, we could check proliferation of cells and also these genes associated. So this we could see, but developmental biology is, I'm sorry, I cannot
it's just that in developmental biology with smaller critters, that you do like light sheet microscopy over time, and then you select cells at the time points where you do it. So they're a little further than mouse work. Yeah. And I'm sure mouse work will get 4D eventually over time. Yeah. And therefore, it's hard to say what comes first and what originated from that, although you do cell tracing. You just see a common progenitor. But it's hard, in my view at least, and I wonder if you agree, to tell if a cell truly differentiated into this and that branch, or if there was a selection of cells to start with, and this and that branch survived. And that would be fundamentally different in developmental biology. One would be differentiation and the other one is selection.
Yeah, I think in this case, really the phase tracing would be key, right? Because we can really track these cells and follow them in their lineage. So this is also why I think we should continue using these mouse strains, which we can actually perform the tracing. Of course, we want to switch more to the more patient side of things, also patient samples. But in our work, we see that it's very difficult to really pinpoint cellular origin, and I think phase tracing, maybe also dual genetic phase tracing models can help with this. But yeah, I would definitely recommend that we keep on doing these phase tracing models. Yeah.
Candidatus, thank you very much. I can think of nobody who would be better suited to do what you just proposed. So go on. Thank you.
Thank you, Professor Janen. The next opponent is not here in this building, but he is online. Dr. Caroline Cheng, welcome. You are Associate Professor of Vascular Surgery at the University Medical Center in Utrecht. Dr. Cheng. Please unmute your microphone.
Oh.
Question, because we missed the first part.
Of your question, which is regarding, of course, the phenotype that we observe in our heart ratio TB analysis. And I agree with you that indeed in the MyH11 mice and also the PGF receptor beta mice, we see less of a phenotype. In our studies, due to limits or constraints, unfortunately, in our mouse breeding colonies, we were unable to ensure also that the biological sex was evenly distributed, especially also in these PGF receptor beta TUC hearts. These were a little bit more dominated by female mice, and this also induced a slightly lower phenotype that we observed there. In the MyH11 mice, yeah, these anyway showed also a lower phenotype. I mean, this was done only in male mice. Yeah, so this is a good question. I don't really know why this phenotype is really lower, right? In theory, the Cre driver should not interfere with the function of these cells. I think this is just a matter of how things went and how the data turned out to be in the end. Yeah.
So you would say it is.
I mean, after submitting this paper and we got some reviewer questions, we also were able to expand a few of these Cre drivers, so the CDH3, the endothelial cells, and also the GLI1. We were able to add another time point. Unfortunately, we were not able to do this for all of our mouse strains due to constraints. You know, the breeding was at that time very difficult, also during the lockdowns we experienced. But yeah, I agree. If we would have more mice, then for sure we would also see a phenotype there. I mean, if you look at the histology also next to this in the periosteum staining, you do see like an increased fibrosis, right? So for sure there is a phenotype. However, it's not reflected in the hypertrophy of the heart in this case. Yeah.
Yeah, I agree with that.
The question, indeed, we were very much interested in this cluster. Unfortunately, there was, I think still there's a little bit, there's a lot unknown about the contribution of this cluster in heart failure. We have performed also stainings in our samples using in situ hybridization to really confirm that this is really present in the heart and not like a isolation artifact. And we anyways always included these cells in our analysis, right? Also in the in figure afterwards, where we figure, what is it? Figure 5, where we try to find like this more general origin of these ECM fibroblasts. We also included there these interferon fibroblasts. So we always considered them. We found it difficult to really associate like a functional meaning to those cells in our study. But yeah, since we anyways we published our data set and all the extra information, so hopefully in the future we can further investigate, or somebody else can download our data set and dive into this. So that's also the reason why we made sure that this is easy accessible and public. So perhaps our successors can look into this. Yeah, but unfortunately for us, it was difficult to really assess any functional meaning to this in our model. Yeah.
Okay.
Thank you for this question. Indeed, these ECM fibroblast population is comprised of all fibroblasts that we phase traced, right, and also we analyzed. However, in our composition analysis, we saw that these GLI1 fibroblasts had this were contributed the most to this population, right? So just to be sure that we saw the strongest effect, we picked those cells actually, and we also had these mice available in the lab, so we could isolate them and perform this overexpression. So it's based also on the strongest effect we saw in our single cell data that we selected specifically those GLI1 cells.
Okay, thank you, candidates.
Thank you, Dr. Cheng. The next opponent is Dr. Elham Bidar. He is Associate Professor of Cardiothoracic Surgery at the Maastricht University Medical Center Plus. Dr. Bidar.
Thank you, dear Pro-Rector. Dear candidate, congratulations on this fantastic work, and congratulations to your team. As a clinician, I learned a lot from reading this, and I saw your models, which resemble clinical models with which we work on a daily basis. Your TAC model resembles the aortic valve stenosis, and it was interesting to see that you mentioned replacement fibrosis occurring, and in the clinical model, when you look at patients with aortic valve stenosis and you look at the biopsies from the ventricle, the main type of fibrosis is reactive fibrosis, especially at the endocardial level, and the endocardial cells are thought of as the main driver of that process. In your chapter 3, I didn't see the word endocardial cells at all. So I was wondering, is it because they are not tagged that you don't see them in this process? Or is your model, you analyze your hearts too early, 28 days? I mean, maybe it was not long enough, or is it a completely different mechanism?
Esteemed opponent, thank you for your question and kind words, and I'm also very happy to hear you enjoyed reading my thesis. Of course, we are looking more at the more biological side, and the model we use, the TAC model, is of course a little bit difficult to compare to what happens in the patient, right? The TAC model is more of a direct injury, right? The aorta gets constricted directly, and then the effect is that the pressure on the heart is, very strong directly, right? In the patient, you would observe this more gradual increase of the stenosis, as you mentioned, and this would not be as direct as you might expect. Regarding the endocardial fibroblasts, so we did not strongly observe this in our data. I think there were also some endocardial endothelial cells which we tracked actually. But yeah, so I agree there are very interesting studies also this year in Nature Genetics. There was a very interesting one about the endocardial contribution, also where they ablated these endocardial cells. They could really show that there was a reduction in the fibrosis observed. So it's a bit of a shame that we didn't observe this in our model, but yeah, I think for sure these cells are of great importance. Yeah. Oh. So yeah, we're missing this information in our model, I think. Yeah. Oh.
Yeah, because the reactive fibrosis resulting from that process is reversible.
Oh.
So we are happy to replace the valve and look at the patient 6 months later and see that the fibrosis has decreased.
Oh.
So that's, but still very interesting findings. So then I have a question about chapter 4. It's interesting because you stented the left coronary, left carotid artery for your fate tracing, and then you stented the abdominal aorta for your single cell RNA sequencing.
Oh.
And you conclude about the coronary artery stents.
Yeah, so a little bit of both, right? I mean, the fate tracing was, of course, performed in the carotids, right? I mean, I know that the aorta and in the carotid arteries, they differ a little bit in the structure, right? In the carotids, you might find more elastin fibers, and later in the aorta, especially the abdominal one, you find more these contractile VSMCs there. I mean, ideally, we would also perform the single-cell-seq in the mice that we had for fate tracing, right? I mean, we tried this also. However, we isolated from 6 mice, then the stented aorta, stented carotid, and this in the end yielded like 500 cells in our data set, so this was impossible, right? With the rats, we could get enough cells to make some statements. But I agree, we should be careful a little bit, to directly link this together. But I think there are similar processes, right? I mean, we focus on the endothelial cells and the fibroblasts. These are both present in both locations. So yeah, this is also still ongoing, right? This work we will continue also relatively soon in our lab. But I think we can make some statements about it. And the mechanisms we find, we validated this. I think this is also of interest. So yeah.
Thank you very much. I think I have time for one more question. So, as a cardiac surgeon, of course, I'm the next step after in-stent stenosis of the cardiologist, and then we try to do a bypass surgery in these patients. So my question is, how do you see in the near future the role of your findings in selecting patients for cardiac surgery instead of PCI? Do you think you will find you can characterize patients with enhanced endothelialization potential or in those characteristics? Perhaps a biomarker that would select for us the right patient for PCI instead of CABG and other way around?
Yeah, thank you for this very interesting question. I agree that for the patient, it would be great if we can detect this more earlier, right? I don't know if this is, I mean, you probably know better than me about the kind of imaging tools that you have to visualize the restenosis and see how this is progressing. The markers we have now is that if there's a lot of macrophage infiltration, this gives you an indication of how the in-stent restenosis will progress, right? So I would look more into like the immune markers that you might find in the blood, because the signaling also that we described in our validations, this is I think more locally, right? So this would be very difficult to measure on a patient scale, right? I mean by taking blood or whatever. So I would recommend more to focus on imaging models to see if we can better predict this. I mean in the end, the stent probably has to be replaced or even with a bypass, but if we can especially prevent this the stent thrombosis forming, this would be key. So yeah, this would be my answer for this. Yeah.
Thank you very much. I give the word back to the prorector.
Thank you, Dr. Bidar. The next opponent is Dr. Pieter Vriesendorp. He is Associate Professor of Cardiology at Maastricht University Medical Center Plus. Dr. Vriesendorp.
Thank you. Dear candidate, first off, I would like to congratulate you and your supervisors and team for this tremendous effort and beautiful work. And for the audience, it's also very important work because I really hate in-stent restenosis. I spent this time making a diseased artery look pretty again, and then I come back later and it looks as bad as I left it. And then both me and the patient are disappointed, and we have to go to Dr. Bidar to have their chest cracked open. So if we can avoid this in the future, I think this really helps in our understanding. And I'd like to discuss on the findings in chapter 4. You write about how stent placement disrupts the cells crosstalk between endothelial cells and fibroblasts. And it upregulates some interaction, but downregulates the majority of the interactions. Is this disruption only present after placement of the stent due to its mechanical injury? We place the stent with high pressure balloons up to 20 atmosphere. And is it only at that time point, or is this an ongoing process, months, years, with the stent in situ?
Esteemed opponent, thank you for your question. And I agree, the in-stent restenosis is a very ugly thing that you observe in the patient. Regarding your question, I mean, we took the 4-week time point after stent implantation. This is what we used to measure this. I think this is an ongoing process. One thing to note is that in the phage tracing in the mice, you can see this blockage is very, very large and it's really dominant, right? In the rats, we rather see a more earlier phenotype of the restenosis. So I think there, it's in what we measured, this is still actually ongoing, actually, and I would expect to see this also in the coming months or years to progress. And this disruption in signaling is likely also caused by immune cells really messing up the normal way of how the vessel functions, basically. Yeah. So this is what we then measure using these bioinformatics tools. Yeah.
Yeah, the reason I asked this is because you also mentioned this as a limitation. We don't use the bare metal stents that often in our patients anymore. It's all drug-eluting stents. But after 6 to 12 months, the drug the sirolimus or any drug that's on the stent is completely resolved, and in fact you have a bare metal stent left.
Oh.
Even then, the in-stent restenosis rate of a drug-eluting stent isn't as high. So something happens in those first 6 months that prevents the in-stent restenosis. And then building on that, you mentioned the scaffolds, right? The bioresorbable stents. And we think there are a promising new device. However, our initial clinical trial showed that there was more stent thrombosis. So as you show in your proposition 7, by avoiding Charybdis, we came too close to Scylla and lost a couple of sailors in the process. Do you think your findings can help to improve new prototypes of drug-eluting stents or scaffolds to still avoid Charybdis, but stay away from Scylla?
Yeah, thank you. I mean, this quote from this paper, I think, is brilliant, right? And they wrote an excellent review, and then to come up with this analogy is just great. So in our model, we specifically used the bare metal stents to not have any disruption of the signaling by the coatings that you might find in patients, right? So you get a clear picture. As you mentioned also with these bioresorbable stent, this is more about the part that contains the coating, right? The polymer that gets resorbed where the coating usually is. And these, of course, are a lot thicker than the other ones. And I think that this polymer that remains also is like a driving force for inflammation, and this is also what causes these symptoms in these patients and afterwards. So I agree. So from our data, I mean, regarding Charybdis and Scylla, right? So I think what is really key is that we don't disrupt these endothelial cells, right? That they cover these stent struts and actually focus then later, maybe even with a slower releasing drug target, that we also keep these vascular cells at bay, and that we keep them in the vascular region, and then that they don't migrate anymore. So this is what I would recommend, like maybe like as I said, a slower releasing compound. Yeah.
Or maybe a drug that's an EPHB4 inhibitor on the stent. You think that will work? Receptor inhibitor, sorry.
Yeah, or like EFNB2. So EFNB2 is the ligand, right? You can also get an antibody blocking agent, right, to capture this this ligand so that it doesn't bind and doesn't activate the fibroblast. So these two suggestions could potentially work. Yeah.
All right. Thank you for a good answer. Do we have time for a short question? All right. This is a bit of a lighter touch, but this is on proposition 11, on your failure to become Jimi Hendrix. Do you want to be Jimi Hendrix, or who is it that you're trying to achieve to be?
Thank you for this also interest in the music. I mean, I think John Mayer has been one of the recent great guitarists, and I think also his playing has been very instrumental in the recent guitar world. Of course, Jimi Hendrix was more experimental in his approach, but in the end, as long as you can make a great song, everyone is happy, right? So
all right.
A mixture of both would be nice, yeah.
Thank you very much.
Thank you, Dr. Vriesendorp. The next opponent is Dr. Armand Jaminon. He is Assistant Professor of Biochemistry at Maastricht University. Dr. Jaminon.
Thank you, Mr. Prorector. Dear candidate, also I would like to congratulate you with your impressive thesis, and I also want to congratulate your promotion team with this thesis. I'm actually very proud that I see you standing here, because I know you still from as a student in our group. When you started doing your work, and seeing you climb up this ladder, now defending here, makes me quite proud. All right. I was intrigued by your statement 12, and I want to ask one of the paronyms, please, to read it out for us.
Opportunities are not always easy to recognize, but when you see one, seize it. Do not wait for the perfect moment. Take a chance and trust in your abilities.
Thank you. So my first question about this would be, whose statement is it? Is it your personal statement, because it's not quoted as someone?
Stephen Fonen, thank you for your kind words. I'm also very happy to see you sitting there and that our academic paths cross once more. This is a statement that I wrote down. This is also why it's, at the end you find my name as a signature, so this is one of my own.
So then my question would be, what is the opportunity that you seized?
I think this is anyways more general in life, you know, sometimes something comes in your path, you go for it, and usually you can plan things out very well, but sometimes life has other plans for you, and I think it's best to stay a little bit more fluid in there and go a little bit with the flow also. So yeah, this is my life vision maybe.
Yeah, nicely said, I think, nicely said. So then I want to talk a bit more about science, and especially now about chapter 4. And there you use, or you mentioned the use of aspirin during your treatment in the mice. And I was just wondering, does the treatment of aspirin itself affect the readout that you found afterwards?
That's a very good question. So during my first year of my PhD, we started with this model, and we were actually struggling quite a lot. We had some mortality rates. We switched basically two things. So first, we administered the aspirin via gavage, right, which was also quite stressing for these mice. And later, we switched to adding it to the drinking water, actually, and this actually solved at least our experimental issues. Our dropouts really reduced quite a lot. The aspirin is, of course, anti-thrombotic agent, right? So, and of course, this is very important in the field of stent, as you know, especially in the early phase with platelet activation and immune infiltration. This can affect this. However, you know, since this is also a very artificial model, we don't want these stents to directly completely clot, right? Otherwise, our model would also not work. So. I think it would influence the more immune and platelet part of the story, but not the actual migration of cells from the vasculature and also the perivasculature.
Yeah, so it's interesting that you mentioned this, because in the rat model you actually don't use the aspirin, if I got it correctly.
Yeah, we use the clopidogrel. We use in the food actually. This is administered via the food actually. This was a model that was designed by also our collaborator, Felix Vogt in Aachen, and he had this established. So we actually collaborated with him. He has great results with this. So I would say it's a little bit different administration in both models that we had. I mean, it's always difficult to adjust these protocols and streamline them, especially with our animal facilities in Aachen. But yeah, so there's a slight difference, yeah.
Could this difference in treatment then also reflect what you found as population? Because I think you already mentioned in the rat population, you find a much more inflammatory part of cells that is also contributing to the restenosis.
No, I agree. I mean, in the mouse model, we only phased the vascular cells, right? So we kind of, I mean, I stained also on these samples for immune cells, and we also observed them quite strongly. You can see in the supplements these pictures, right? The T cell and also the macrophages. And not by phage tracing in the rats, we could not do this. We captured everything, right, which include a strong immune response. We also see this in the stented samples. The immune population expand greatly. But we also capture the vascular cells there. Yeah. So I hope that answers your question.
Yes, it does. Yeah. Thank you. So then, related to this, could you think of other treatment options that you could have chosen that have a similar effect, so an anti-thrombotic effect, that maybe would have been better, so that you could still keep the inflammatory response in there as well? Especially, I think, in the mouse model, this would be of interest.
I find it difficult to mention one now, but I think, on the other hand, you don't want to disrupt this process too much, right? Because we want to simulate what happens in the patient, and the patient also, I mean, they use dual antiplatelet therapies, right? So maybe something similar to what we use there, but just to keep the picture a little bit the same. If we start, you know, messing up all these pathways, then the data that we get might be not represent actually what's in the patient. I hope that satisfies your answer. Your question.
Fine. Yes. So then, I think your predecessor has shown beautiful studies, and I want to maybe go a bit further into looking into the treatment. And then, I mean, it's not really treatment for inpatients, I think, for as far as we are now, but I miss a little bit the ablation part. That is beautifully shown by your predecessor, like I said. Did you try this? And are there some highlights that you could give us that as of results that were shown there?
Yes, actually, a very good question. We attempted this with these stents in mice. However, this was unsuccessful somehow. Also, mortality was quite high. So we tried it, and in the end, we did not really see a difference. So yeah, it's a challenging model, the IDTR model we used for this, specifically also for GLI. Yeah, but yes, so unfortunately, my supervisor has shown in other fields, like in also kidney fibrosis, that if you ablate these cells, you strongly reduce the fibrotic response, right? And also, I think this would be similar in our stent model. However, yes, for some reason, we're challenging quite a lot with this. So I agree, we should further look into this to also strengthen our claims that we have here. But at the moment, yeah, so this was still ongoing, I could say. But yeah, was unsuccessful in our hands, yeah.
It's a difficult model to tackle, I guess. So maybe a short question last. I saw that you mentioned some synthetic markers, which intrigued me, especially I think you mentioned them for the small muscle cells. Could you explain me a bit of what they show or what they represent as molecules? Yeah,
so we indeed measured like some also some genes that are associated with this vesicle secretion. This is TSPAN and this CRYAP-B. So these we observed in our data set, and you probably also know, right, from our vascular calcification studies, these are, of course, very much involved, right? Also, maybe the communication with also platelets. I thought it was very interesting that we found these cells. This is also why we reported it. However, it's still difficult to say how it would affect specifically the in-stent restenosis. I mean, it's of course more known in vascular calcification, but it's here still ongoing, I would say.
Mr. Halder, you understand that the time appointed for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis and in particular quality of your defense, and then please await our return here. Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. I know what's on the line. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even Even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Meneer Halder. The degree committee here present has assessed the quality of your thesis and in particular the quality of your defense today. In view of its positive verdict and of course taking into account your previous qualifications, the committee has decided to grant you the degree of doctor. And Professor Schurgers is authorized to confer upon you this academic distinction in accordance with Dutch university custom and law. I invite now your supervisor to take the floor. Professor Schurgers.
But before, I have to ask you something. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Maurice Halder, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of your committee and affixed with the official seal of the university. Dear Dr. Halder, dear Maurice, I'm the first that is allowed to congratulate you with this fantastic achievement. And in these congratulations, I also want to congratulate your supervisor from Aachen, Professor Raphael Kramer, with whom I have a very fantastic collaboration. Also want to extend my congratulations to your family and friends, and to your girlfriend. Congratulate, maybe you have him back a little bit more now. Now the hard work is done. I also want to thank the members of the thesis assessment committee and also here in the opposition present today. Especially I want to thank Professor Willy-Janen de Gent from Aachen, traveling all the way, my long-standing collaborator. He was even present during my PhD defense, and that is already a long time ago. Dr. Caroline Cheng, thank you for being present online from Utrecht. Thank you for participating, and also here the members from Maastricht. Dear Maurice, number 15, ESR number 15, early stage researchers in a European Horizon 2020 ITN consortium termed IntriCare. This collaboration between Maastricht, Stockholm, and Aachen was initiated already a long time ago. I think it was in 2016 that the contract was signed, and 2017, I think the actual practical start of all the ESRs was done. And within IntriCare, the aim was to investigate the role of vascular remodeling and atherosclerosis in the setting of chronic kidney disease. IntriCare was a network, or is a network, of colleagues and friends. And they were all willing to collaborate and to contribute and put effort and energy, which forms the basis of a success for such a Horizon 2020. And you were clearly part of that. These 15 ESRs were selected from all over the world. Germany, England, Greece, Poland, Romania, Russia, Brazil, Pakistan, China, India, and last but not least, the Netherlands. A wonderful group of young researchers. But to select these candidates, one had to go for a PhD student that was not trained within their own country. So I couldn't choose you. Back then, you were a very talented first bachelor student in our group, and later on a master student. You did your master thesis in our group together with Dr. Jaminot. And you did this very successful. You were hardworking and very smart. You won actually the Unilever Thesis Research Prize in 2017. And this prize needs to be named. It was awarded to outstanding postgraduate students in the Netherlands. And from each of the 13 Dutch universities, one master thesis was selected. And from that, there was one winner, and that was Maurice. And this prize was to strengthen the bond between industry and academia and to motivate students to excel. Well, clearly you did, and you proved us today. Maurice's thesis was focused on the effects of arterial remodeling and vascular calcification in the development of cardiovascular disease. So it completely fitted in the aims of injury care. So it was clear to me that Maurice had to work in academia, had to work for a PhD career, and actually stayed in our consortium. So it was obvious I recommended him to my close collaborator, Professor Raphael Kramann, in Aachen. And with that, I give you the word for the final part.
Yeah, ladies and gentlemen, esteemed colleagues, students, friends, and most importantly, our distinguished guest of honor, Dr. Maurice Halder. Today is a day of celebration, reflection, and achievement. We have gathered here to mark a significant milestone in Maurice's life and academic journey, his PhD defense. This momentous occasion is not only a testament of Maurice's dedication and perseverance, but also a tribute to the support and encouragement he has received from his friends, family, and mentors throughout his long and challenging process. Maurice's journey towards earning his PhD has been filled with countless hours of research, experimentation, late nights, early mornings, and including many card games and drinks in the PhD office. You did a fabulous job in tracing all kinds of cells in kidney, heart, and vascular disease models, and are the biggest confocal imaging expert in the lab. In addition, you also built an outstanding expertise in single-cell RNA sequencing of cells, fact sorting, writing papers, and all kinds of complex molecular analysis, and not to mention the most important skill, making the best memes for everyone in the lab and for wallpapers in the PhD office. One meme I still remember well is Saving Private Kramann, using the theater poster of Saving Private Ryan. Getting a PhD is often likened to a marathon and test of endurance, patience, and resilience. Maurice, you have not only completed this marathon, but you have also crossed the finish line with flying colors. Your determination and passion for your research have been evident from the very beginning, and today we stand here in admiration of your accomplishments. During his PhD journey, Maurice has not only excelled academically, but also demonstrated leadership, mentorship, and commitment to giving back to the academic community. He has been a source of inspiration and guidance for his peers, and his willingness to share his knowledge and expertise has enriched the experience of those around him. But Maurice's success today is not just a personal triumph, it's a reflection of the collective effort and support of many. We must take a moment to acknowledge the family and friends who have stood Maurice throughout his academic journey, providing the emotional support and encouragement that is so crucial during the challenging moments. As we celebrate Maurice's achievement today, we are reminded that a PhD is not just a symbol of knowledge, but a commitment to lifelong learning and the pursuit of excellence. It is a responsibility to use knowledge and skills to make a positive impact to the world. Maurice, as you stand here on the precipice of just received your doctoral degree, I encourage you to continue pushing the boundaries of knowledge, to remain curious, and to use your expertise to address the challenges facing our society. The world needs thinkers and leaders like you who are not afraid to explore the unknown and seek solutions to complex problems, including, of course, also memes. I'm glad that I will still be working in tight collaboration with you in your new position at our spin-out company, Sequantrix, where you will be developing urgently needed antifibrotic therapies for patients with kidney, heart, and bone marrow failure. In conclusion, today is a day of celebration, not only for what have you achieved, but for the promises of what lies ahead. Maurice, your journey has been a testament to your dedication, resilience, and passion for your field. As you move forward, may you continue to inspire others with your work, and may your future endeavors be filled with success and fulfillment. Ladies and gentlemen, please join me in congratulating Maurice on this remarkable achievement and in wishing him all the best in his future endeavors. Maurice, the world is now yours to explore and make a difference in. Congratulations.
Thank you.
Esteemed Dr. Halder, dear Maurice, it's my great pleasure to congratulate you with your doctorate, and my congratulations also are on behalf of the Board of Deans and Maastricht University. And I would like to share some impressions of the committee with you. We have heard and seen a very clear presentation, and I think you have, and the committee thinks that you have succeeded in make your scientific work very clear to lay people, and that's exactly the point and the purpose of this kind of presentation. Your thesis represents great work. You did outstanding quality studies, well published in high quality journals, and I think that you have performed a very readable thesis, for many people relevant. Your defense, well, we have seen that your answers were in detail both on the work that you have performed, but also in terms of the future, what has to be done, or what could be done by you or others. Your defense was very adequate, very clear, complete, and you also translated many aspects from bench to bedside, and I think that's important for this type of studies. We are very satisfied with your defense. I would like to include in my congratulations your two supervisors here opposite, sitting to me, Professor Schurgers and Professor Kraman. Congratulations with this excellent research that we have seen today and that you have supervised for 4 years, longer? Longer. Of course, I would like to include in my congratulations your family, your parents and your grandmother and your partner and other members of the family. You may be proud, sitting here on the first row, you may be proud on his effort and what we have seen in his thesis and defense today. I also include in my congratulations your friends and former colleagues and still colleagues that are still your colleagues at the present. So congratulations all that have contributed to your studies. And I wish you a successful professional career and a happy personal life, because that's also important. I would like to thank the members of both committees, the Thesis Assessment Committee and the members here of the Degree Committee. Thank you for reading the thesis critically, to have nice questions, interesting questions, and an interesting discussions. And I would like, before I close the ceremony, I would like to invite Dr. Cheng, who is online present here, to congratulate the young doctor. The floor is yours.
Thank you. So, dear Dr. Halder, dear Maurice, I congratulate you and your family. And of course, also your supervisors with your impressive accomplishments. I had a very good scientific discussion with you today. In fact, I only realized that you could only hear half of my first question when I went back to the breakout room. Despite of this, you were able to somehow still able to provide me with the right answer. So I found it very impressive. Reading your thesis was a pleasure. And I believe that based on what you have shown in your papers and the way you've conducted your defense today, that you have a very bright future in science, in particular in the cardiovascular field. So I hope to bump into you in the future and have many more meaningful scientific discussions with you. But for today, please enjoy and celebrate. You have definitely earned this moment in the limelight. Congratulations.
Thank you. A couple of announcements to make. We will make a picture here with the online member on that screen, because that screen is not working for that. Sure. So we are having a picture with the committee, and that your online opponent is also in the picture. And I would like to ask you to go to the place for the reception, which is in the rafter, and the beadle will show where that is. And I would like to ask the Close family to stay here, because we also take a picture in the hall on the stairs. So the people on the first row may remain sitting here. Hereby I close this academic ceremony.